This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 247, recorded on July 22nd, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How you
doing? Are you good?
Fine.
You're fine? Very good. Glad to hear it. Also joining us from northern Michigan, Michele Swanson.
Hello, everyone. Yeah, it's been a pleasure. We call it up north
in
Michigan.
Up north.
Yeah, it's been a pleasure to be able to spend more time outside.
Yeah, you walk around, you go on hikes?
Yeah, hikes and a little bit of paddleboarding and a little sailing. And
nice. Watch
kayaking.
Out for the ticks and the mosquitoes. You have any up there?
Oh, yeah. You bet we do.
But it's not really hot, right? It's kind of coolish.
No, it's pleasant, like 70s, and then it cools down to the 50s, 60s at night. Oh, and I've been golfing. I can't forget that.
Where do you go to golf? You must have to drive, right?
Oh, there are a lot of public courses around here.
Nice. All right. And finally, from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
You don't golf, do you, Michael?
No, I golf once every 5 years when my brothers come to visit, and they need someone to lose.
Okay. Well, you know, folks, summer is zipping by. It's almost the end of July. Can you believe it?
It's shocking.
It's just, it goes really quickly. Every week just zips by, and it's almost August, and pretty soon we'll be...
Don't say it.
Okay. We just should enjoy what we have, right?
Yes,
right.
Each day.
All right, let's get into some microbiology here. Let's start with a snippet from Michael.
So the title of the paper that we're going to talk about today is Gut Microbiota Through an Evolutionary Lens. It's a perspective paper that appeared in the April 30th issue of Science, the one with the cover Pandemics in Waiting, which I've been reluctant to read for fear it would keep me up at night. And so I finally broke down and opened it up, and I spotted this paper. And this paper is by Carmody, Schaechter, and Rees, and they're from Harvard. And the paper is not necessarily our normal fare for a snippet, but it's one that I believe is worthy for you all to consider as part of your summertime reading or deep thinking as you sit on the beach reading that most recent Andy Weir's book, Hail Mary, which has a microbial spin to it, thinking about the wonders of the microbial world. And I also encountered this paper after stumbling into Gina Colada's column on Monday in the New York Times, You're Missing Microbes, but is rewilding the way to get them back. And that's in the online edition of the New York Times from Monday the 19th. And her piece starts, innocently enough, with the following line, quoting, "As the sun set in Tanzania on a September evening in 2014, Jeff Leach inserted a turkey baster filled with another man's feces into his rectum and squeezed the bulb. The feces, he said, came from a hunter-gatherer who was a member of the Hadza people and lived nearby. We'll learn from her story that the Hadza people are thought to harbor a form of the primordial microbiome of humans. If you will, the Hadza produce real-time paleofeces. So I challenge you to see how many times you can work that word, paleofeces, into polite conversation. But while the concept of rewilding, or the equivalent of, if you will, rebooting our microbiome to its original default software, if you will, to give us better health, is a topic unto its own. And that's what the paper by Carmody, Schaechter, and Rees really force us to think about how to design experiments And it's really something from the final line of their paper, if you will. This is the nano snippet. There will doubtless be scenarios where gut microbiota restoration will improve health in industrialized populations. But consideration of the sources of mismatch and their complex dynamics is necessary before pursuing targets for intervention. So this one line should give us all a hard stop in our scrounging the utensil drawer for the turkey baster or scouring the internet for paleofeces, or even whether or not many of us are taking probiotics. So how did the authors reach that conclusion from the nano snippet? That's principally contained in the one figure associated with this perspective. And it's this imagery that they use to describe the dynamics of our industrialized gut and how the industrialization that the human race has undergone over its evolution has altered the human host as well as its microbial and our environmental interactions. And that's effectively what I want to challenge you, our listeners, to think about from today's snippet. This will be an old story to anyone trained in evolutionary biology, microbial ecology, or even population biology, where one considers, often with complex mathematical formulas, understanding how and what perturbations can do to influence population dynamics, or if you will, why some societies thrive and others fail, simply how changes can influence the fitness of the community. Now, the imagery in that figure they use to describe this concept is nothing more than an equilateral triangle. And as its name implies, is used to describe the ideal human microbiome environmental condition. So in their model, they are effectively getting us to contemplate Voltaire. Perfect is the enemy of the good. So in their model, the equilateral triangle is the ideal phenotype, and they operationally or arbitrarily define that as health. And there are no incompatibilities in that perfect triangle with respect to health. The microbiome is complete in sync with the environment and our genome. And as the human condition begins to perturb itself away from the best fit or the perfect equilateral triangle, or where the area of the triangle moves from the perfect to an imperfect state, from the perspective of our definition of health, the length of the vertices of the triangle are altered. If you will, the lines get longer, resulting in a change to the shape of the triangle, the angles within the triangle. And so all of that is used to suggest what the input is doing to the principal phenotype that is being measured.
Michael, can I interrupt you?
Sure, please.
I was not as enthusiastic as you are. I don't think that triangle means very much or that it adds anything to our knowledge. I mean, where's the meat?
Yeah. Well, the meat is what I'm coming to. And
it describes the interrelationship of the three, that no one acts independently, is how I interpreted that figure.
And that is absolutely correct, Michele. And they use a term that I hadn't heard about since my undergraduate days working on cave spiders. And that's canalization, which is the measure of a population to produce the same phenotype, regardless of the variability of its environment or genotype. And this is the concept, or the meat. It's a form of evolutionary robustness, sending us back to this equilibrium of health. So with the phenotype that they are operationally using as health, you get the idea that their outcome is going to be hard to measure for what defines health. No cancer, no obesity, no diabetes, but without those, we have much shorter lifespans, or the trade-off of a long lifespan with comorbidities of coronary artery disease, stroke, diabetes, etc. So the shape of this triangle, or the sides of the triangle, in their explanation of the effects of industrialization on human health, serve to illustrate and help people to think about the degree of health-relevant incompatibilities. They reduce the problem to the following statement. When is it better for the gut microbiota to track versus resist environmental change? Implicit to the benefits of rewilding that was offered in the Collada story, where Mr. Lynch was trying to reboot his microbiome to a paleo microbiome, going back. But what they point out in this perspective is Lynch was banking on the fact that paleofeces can accurately be characterized, and that such a microbiome, the paleofeces, can promote health in an industrialized human environment, and that any such microbial manipulations that we do will have predictable phenotypes in an outbred population like the human race in our industrialized population. So Carmody, Sarkar, and Rees offer as examples to help us think about this or illustrate this dilemma. They offer us that the fiber degrader, Prevotella copri, whose number is often enriched in guts of individuals in non-industrialized communities like the Hadza, has shown both benefits for glucose tolerance, but at the same time, the propensity to exacerbate chronic inflammatory conditions. So depending on the context, more fiber, less type 2 diabetes, but more irritable bowel syndrome. So consider the cancer-fighting properties of non-industrial microbiotas. Recall that such microbes like Prevotella generally enrich the host capacity to ferment fiber into these short-chain fatty acids that cover diverse metabolic and immune benefits. But what they want us to appreciate, and they use examples that we've already talked about some on TWiM, like the contribution that short-chain fatty acids from Akkermansia can contribute to Parkinson's disease. These short-chain fatty acids can lead to context-specific developmental trade-offs with anticipated pleiotropic effects like weight gain through their ability to increase energy salvage, and as I said, increase the progression to Parkinson's. And similarly, the Collada article talks about the two twins out of a manuscript that was published a number of years ago out of Washington University, out of St. Louis, by Jeff Gordon's group, where we had twins, and one became fat from a microbiome transplant and the other did not. And so these two individuals could exhibit divergence responses when you consider rewilding of the gut microbiota is dependent upon idiosyncratic factors such as parasite and viral burden that you may have had over your life that are then in turn involved in immune training and various other gene environmental interactions. And the rewilding proponents that Ms. Collada introduces us to in her fascinating story make the argument that there was a time in our evolutionary past where humans were better matched to control the two sides of the triangle, namely the environment and the microbes, simply because we couldn't manipulate our environment as easily as we can today, nor did we have many food choices. So as we have moved from hunter-gatherers to an agrarian to an industrialized populations, the lines of this triangle have not always moved in sync. So again, Carmody and Sarkar and Rees challenge us to ponder to the extent to which the human race has been able to canalize, or the ability of our population to produce health, regardless of the variability of its environment or genotype, namely how to stay healthy, is likely a function of our ability to manipulate the triangle through medical intervention. And they offer that the burden of infectious diseases has generally decreased in the human population, while the burden of non-communicable diseases has increased, which led me to propose the following Talmudic question to Elio, who thought it was too easy. And the question was, how has the burden of infectious diseases generally decreased, while the burden of non-communicable diseases increased in the human race during the latter half of the 20th century? However, here's where Carmody, Schaechter, and Rees challenged the microbiologists to look at the data beyond the obvious answers of sanitation and antimicrobials and public health by offering us that the industrialization of the human race may be providing us with an evolutionary lens with which to help us appreciate that complementary hypotheses that advance our understanding of why human microbe interactions have responded in the way they have to these perturbations as a consequence of industrialization. And again, I just want you to ponder this as you're sitting on the beach, thinking about the fact that microbes have genes for metabolizing complex carbohydrates, but those genes in our microbiome are waning in the industrialized corn-sweetened everything diet that we consume, and offer us that the phenomenon of the unfortunate loss of host-adapted microbes capable of metabolizing the unusual carbohydrates that we were forced to eat when we were hunter-gatherers or early agrarians would be viewed in the evolutionary lens from the perspective of metabolic streamlining, where if you don't use it, you lose it, sort of like me in the solving of differential equations. Things more important for the current environmental circumstances are retained. And one of the most telling pieces of the data they offer us comes from the perturbations from the microbes that they studied in the Hadza, where seasonally the Hazzas lose these diverse microorganisms as they are seasonally driven, where they've already disappeared from our industrialized population. So summing this all up as you sit on the beach, reading the newest John Grisham or Andy Weir, is to consider that competing fitness interests and the higher plasticity of the gut microbiota versus the human host and our environment is a ever-present condition, both in the past as well as today, with the variable and sometimes unpredictable effects on human health. And Carmody and Schaechter challenge us at the end to think that the most effective way to manipulate the gut microbiota in the service of health may be the challenge that we have to disentangle which aspects of health are promoted by matching the microbiota more closely to the host or to the environment, or to a lesser extent to both. In other words, how to drive back to that perfect triangle. And they reduce it to a one-liner to end. It's clear that restoration will require a scalpel and not a sledgehammer approach to do this. At the end of the day, the concept is canalization, the ability to have a robust phenotype to restore ourselves to health. So that's the snippet.
I'm sitting at the beach and I'm getting sunburned.
Well, that's what you're supposed to do at the beach.
I'm sorry, I do not share your enthusiasm. This is all sort of a general, broad way of phrasing the question. But there's no meat. We don't have enough data. This is all an anticipation of the day when we're going to have the data. Until then, why are we talking about this? I don't see it.
Well, I'd
I'm
say
sorry, Michael.
I'd
I
say
don't
that
see it.
the Carmody commentary really is trying to make that point, that there are so many things we don't yet understand and assumptions that are made that are really not based in evolutionary principles. For example, the one that I found most sobering is the reminder that the strong selective pressure is for reproduction, not for the ability to run a 5K race without pain when you're retired.
Yes.
Yep.
Right.
Okay.
So
That's right.
And that's absolutely it. And my challenge and why it's beach reading is to tell people we need to design the proper experiments with good controls, with a large enough sample size, and not done in inbred C57 black mice.
well, and also just the idea that you could take an ancient let's say a feces from a population that is not industrialized and put it in yourself and have it even take hold. I mean, we've got colonization resistance that's a really robust ecosystem in our gut, and they're not going to be able to establish a foothold based on what we know right now.
Yeah. I mean, I think there are, as Michael pointed out, there are aspects of the gut microbiome that maybe could be improved, but to think that an ancient one is going to just be generally better
is not it.
I think this guy was crazy to do what he did, and maybe he's going to get some other infection, and how does he, some other virus or parasite? Right. That's crazy. But do we really think that one day we want to know what a good microbiome is and everyone should have it? I don't think so. I think there are other issues that are more pressing. I think I want to quote Michael Pollan. He said, "Eat food, not too much, mostly plants." And I think that solves everybody's problem, really.
Right. And actually, that's where the microbiome field is pointing as well. More fiber, less simple sugars.
I mean, I think it's worth thinking about. It's an interesting idea, right? But we're here. We did this, and we have the microbiome that went with it, and we're not going to go back. Sorry.
Nope. No turkey basters in my future.
But I think we should figure out how it goes in hand with other diseases. That would be very nice. It's hard.
And diet.
Would be helpful.
It would be very good to do, and I'm sure we're going to see a lot of work in the next 50 years. So
yeah,
that's quite interesting. But I think eat food, not too much, mostly plants. There you go. I don't really.
That's the title.
I don't really. I guess I do eat mostly plants. Yeah. Especially with our garden. We have a great garden now, man.
Yeah, how's it going? How's your garden growing?
Michele? Do you garden?
I do not, but tell me, how is it growing? Are you getting, what are you harvesting these days?
We built raised beds over the winter because everything was on the ground last year. So so far we've harvested quite a bit of lettuce, potatoes. The harvesting of potatoes is now, we got tons of potatoes, red and white potatoes. They taste very good because they're right out of the earth. We're getting cucumbers now. The tomatoes are still green. The squash is growing like crazy. But this year, because it was hot, the lettuce did not do well. The lettuce and the beans. And we have eggplants growing also. So
wow,
it's fun. It's a lot of work, though. Having a garden is a lot of work.
It
is.
What is that, a tomato?
That's a tomato.
Already? I guess because you're in the South, right?
Yeah.
I guess there's something good about living in the South, right, Michael?
The tomatoes come up. It didn't get. This is supposed to be a big boy, but it's not very big.
No, it's not big.
And it's only because I grew it in containers. I don't like to use pesticides, and we have those caterpillars that eat the leaf of tomatoes.
So,
soap works.
We've got another really cool story that Vincent's going to tell, and I can't wait to profile the three first authors.
All right, so this is a Nature Medicine brief communication. Potent antibody-mediated neutralization limits bacteriophage treatment of a pulmonary Mycobacterium abscessus infection. And I saw this, and it's right up my alley. It's got viruses. It's got antibody neutralization. So I thought I should do this. And the first three co-authors, Rebecca Diedrich, Krista Freeman, and Jan Nguyen. And then the last two authors are Graham Hatfield and Kira Cohen. This comes out of the University of Pittsburgh. and Johns Hopkins. So this is all about phage therapy of human bacterial infections, which we have discussed before. As you know, it's being re-explored because of the issues with antimicrobial resistance. And this particular paper is about non-tuberculous mycobacteria, which are becoming an increasing issue. And they have also resistance to antimicrobials. And so bacteriophages are being explored as therapeutics. And not too long ago, a paper was published by this group describing a 15-year-old who received a lung transplant and had cystic fibrosis and disseminated Mycobacterium abscessus infection. So they treated this individual with a intravenous cocktail of mycobacteriophages. And that individual actually did pretty well. However, and in fact, that paper we discussed a bit on TWiM number 44, where we had Paul Young from Yale, who does a lot of phage therapy work as well. But that individual, of course, was immunosuppressed as a consequence of the organ transplant. So he did not make antibodies to the phages. And so they say in this paper, what about in an immunocompetent person? Would the immune response be an issue? And this is always discussed, especially if you're delivering the phages intravenously. Boy, are you stimulating the immune system. So that's what this paper is about. And they have the patient is a single 81-year-old man. He has lung disease, which caused by non-tuberculous mycobacteria. It has been treated. He's been treated for a while with different antibiotics, yet continues to harbor bacterium, continues to have disease in his lungs. So they have an isolate from him. It's called GD82. And they show that it's resistant to a wide range of antibiotics, but they look for phages, microbacteriophages that will lyse this bacterium, and they find three. They have great names, Muddy and derivatives of BP and ZOJ, which were the same three used to treat the 15-year-old lung transplant recipient previously. And I have to say, at least some of these phages come from the high school phage screening program.
C phages.
Right? C phages,
that
right?
Graham Hatfull initiated.
Where for a high school project, they go out in their backyard, they get some dirt, they isolate a phage, they do some genome sequencing, they annotate it, and then they can name it after themselves if they want. And that's where you get, Muddy and Zoe J. Okay, so these three phages will kill GD82 in vitro. However, they do say if you do kill this bacterium with the three phages, you get some colonies that will grow up. They're resistant colonies, but no colony is resistant to all three. Okay, they're resistant to one or two, but not all three. Okay. So what they do is say, let's treat the patient with this. So they prepare a cocktail of three phages, high titer, highly pure, endotoxin-free, sterile. They get FDA approval. They get Johns Hopkins approval. And they continue to treat this patient with antibiotics. But then they give the patient this three-phage cocktail intravenously twice a day, 1 times 10 to the 9th plaque-forming units.
Wow.
That's a lot of phage.
It is. To do that daily.
Twice daily, intravenous, right? I mean, it's not easy.
How much work would have gone into just preparing that amount of pure phage that's safe to inject into a patient?
Well, when we were visiting Texas A&M, where Ry Young has a big phage group and he does some phage therapy, he said in a pinch you can do it really quickly, especially if it's an emergency. So, but it's not easy. So they of course are monitoring this patient. They're doing chest tomography. They're looking at mycobacterial burden, And of course, they're looking for the acquisition of phage resistance, and they want to see if he's adversely reacting. So it's a 6-month treatment, no adverse serious events. They continued it for the entire 6 months, no interruptions. Before the treatment, the amount of M. abscessus in his sputum was 2.4 log CFU per mil, and that went down to 1.2 1 month after initiation of the phage therapy. Unfortunately, they went back up. So 6 months later, it was 2.2 logs CFU per mil higher than the pre-treatment levels. So went up. But if you got those bacteria from him, those mycobacteria, in vitro, they were still fully sensitive to phages muddy and BP, although there was little resistance to Zoe J. So the reason for the failure, basically, of this treatment was not resistance of the mycobacterium.
Like, as we're worried about with COVID-19, is the virus going to change and now, skip out on our therapy? That wasn't the explanation, was it?
It was simply that the patient's spleen was probably filtering the phage out as quickly as they put it in, or they didn't give them enough phage to find all the Mycobacterium abscessus that was hidden in cryptic sites within his body, and then it went back to reseed the particular
those
areas.
are possible explanations, but there's another one.
So they end up what they investigated that turned out to be correct. They look at the antibody response. Of course, when you put a foreign immunogen into anyone who's immunocompetent, you'll make an immune response. And putting a lot of virus daily for 6 months will give you very quickly an immune response. And they say before phage treatment, the patient had little or no antibody response to one of the phages, BP, and weak to moderate binding to muddy and Zoe J. That's very interesting. So this patient already had a little bit of antibody to those two phages, which brings the question: how did this person encounter it? Or maybe it was a cross-reacting phage. All right, but this serum pre-treatment did not impair the infectivity of any of the phages. Only very, very low neutralization, despite the fact that the antibodies are binding the phages, but they're not neutralizing antibodies. But a month after starting the treatment, they see increases in IgG, IgM, and IgA to all three phages. And this continued for two of the phages. It went up for 2 to 3 months and then leveled off. But for muddy, it continued to increase for several months. And that's typical of antibody responses. They can be variable. By month 4, IgG to each of the phages exceeded titers of 1 to 1,000,000 dilution.
Wow.
I mean, that's a lot.
Those are efficient antibodies.
Really good antibodies. And so then they looked at neutralization activity, right? They ask if we mix these antibodies with the bacteriophage and then infect cells, can they block infection? And so they have substantial neutralization activity. They can reduce infectivity by 2 to 3 orders of magnitude after only 2 hours of incubation of serum with phage. And longer incubations, you can reduce it even more. At 2 months and beyond, serum was very neutralizing for muddy, reducing infectivity at least 6 orders of magnitude after 2 hours of incubation. That's huge.
That is so impressive. That's great if you don't want that bacteriophage in your body.
Yeah, well, now they have very high titer serum against these phages.
Yeah.
The other two phages, 3 orders of magnitude reduction. And then they go on to look at what these antibodies are recognizing. The main antigen appears to be the tail tube of muddy and some of the capsid subunits, and they also have responses to less abundant proteins. And so what they see is actually independent antibody responses to each of the 3 phages. There's no cross-reactivity. Each of them gave rise to a set of antibodies that interact with proteins of each bacteriophages. So basically, they conclude that this antibody response limited the effectiveness of this treatment for this individual, and so after 6 months they stopped the treatment because it wasn't doing any good. So they continued with the antibiotic therapy, but we don't know what happened to this patient.
Although I can give an update. Go ahead, Elio. You go first.
It follows that if they have done this in an immune compromised patient,
Sure.
the phage would have continued to work.
That's right. Yeah, as it did in the 15-year-old.
So maybe this is the phages are most indicated for people who are infected and immune compromised, and that is unable to mount an antibody response.
Yeah.
I can share that the first author on this paper and also the 2019 paper with the lung transplant teenager, who is Dr. Rebecca Dietrich, she wrote that the patient was recently switched to aerosolized phage therapy.
Yeah.
So recall, this is a respiratory infection, and so they're just nebulizing it into the lungs, and they're hoping that they can then coat those mycoplasma and take them out of the equation, have the immune system clear them before the immune system gets a chance to present the antigen and raise antibodies.
Are they using a different set of phages for that in this patient? I guess
using
so, right?
the same 3 mycobacteriophages.
Oh.
So it's too early to know what's going to happen. But speaking of the patient, I want to share that Jan Nguyen, who is one of the authors as well, just said what an incredible patient they've been working with. He's got his optimism and bravery have been abundantly clear. He and his wife have been resilient and determined in combating this pulmonary series of infections, and his willingness to engage in the experimental therapy demonstrated courage that reminded their team of the privilege they have in investigating translational research.
Yeah, I don't know nebulizing is a good idea, but since he already has antibodies, they're likely to come into the mucosa and block them. But I think that's not a bad idea, although you will develop secretory antibodies as well. So,
right.
They actually say that the immune reaction is going to be a substantial impediment to phage therapy, especially given intravenously. They are actually surprised that resistance to the phage is not an issue, which is good because it could have easily selected, right? Because they treated for 6 months. But maybe the cocktail of 3 is a key to that, right?
Yeah, one question I had is, there are other clinical scenarios where we intentionally suppress the immune response, like when a person gets an organ transplant. And could they do a short-term immunosuppression to try to knock down the infection and then hope that when they withdraw the bacteriophage, then traditional antibiotics might be sufficient?
Yeah, that's tricky because they're infected, and then to immunosuppress them is kind of
right. Yeah, you've got to have a lot of confidence in your bacteriophage. That they'll do it.
It's interesting you say that they've switched to nebulizing, because they say in the paper we initially chose intravenous because he had his type of lung disease would potentially limit the effectiveness of nebulization, but maybe they decided to try it anyway. Obviously they did,
Right, and it's the senior author, Kira Cohen, is the physician who is managing these cases, and each of the 3 authors just spoke to her commitment and devotion to her patients, and really this project could not have been possible without her. Her leadership.
One of the suggestions the authors make is that maybe we should do serial treatments, 1 phage, a second, and a third, instead of all 3 to get antibodies. But then you're going to get resistance, I think, right? That's the problem with that. So it's not easy. It's not an easy solution here to figure out.
Do you think it puts a general crimp on the idea of phage therapy?
Well, I do, but
to my mind, yeah.
Maybe what do you think, Michael? Having worked on this, right?
We worked on this, and the area that we focused on is engineering our phage so that the immune system would not clear them. And you can actually pass phage through the immune system successive number of times, and you can isolate out phage that are resistant to clearance. And but it requires that you passage it many times, and we were able to isolate such phage that were resistant to immune clearance. And the other question I had is, did they calculate how many phage binding sites, and did they adjust their dose accordingly to account that there were sufficient phage per the patient's mass in order to dose the infection properly? Over 20 years ago, we published a paper on how to calculate the number of phage to give to an individual to clear an infection, and it's languishing in the literature from once upon a time.
I'm not sure they did that. I mean, it doesn't mention it in the methods. All they did was, do susceptibility assays at various points, but not that particularly. So, Michael, when you selected for phages that had resistance to antibody development or clearance, which one?
Clearance. We did the whole kit and caboodle. We specifically put them into the animal, and then we just isolated sera and said what phage are left.
Oh, okay. And what changes are associated with that?
We never got to the structural characterization because we couldn't find a patron that was willing to fund those studies.
The problem with that, of course, is that it adds to the length of time to tailor the therapy for the patient, right?
Right. Because
as it is, you have to get the isolate, you have to find phages that will kill it, and then in your case, to make them antibody resistant is really hard.
And in fact, the most successful phage therapy group out there, the folks from Tbilisi, Georgia, they typically use phage cocktails to treat their infections, and they just make a very large titer of phage and to try to overwhelm the system and hope for the best to see if you can clear the infection. I think part of the problem here is that Mycobacterium abscessus is not a fast-growing microbe from the perspective of a phage. Mycobacteria are notoriously slow growers when you contrast them to the typical infections we see in patients with this disease profile, at like Pseudomonas or E. coli or one of the KPCs. And so consequently, the phage need to hang around in order to kill newly emergent bacteria that arise out of the monocytes or the non-professional monocytic cells that happen to be housing these bacteria. Because again, mycobacteria are facultative intracellular parasites.
Yeah. I think that one approach might be you need to have a whole slew of phages, and you try 3. When you get your antibodies, then you switch to a completely different set of 3, and maybe you can engineer the capsid so that it's different enough. So we do that with adeno-associated virus vectors just to get around this issue, and maybe that's something they're thinking about.
Yeah, and they're easy to manipulate, I would hope. Huh? Vincent, we know enough about these bacteriophages?
I think so. We know where the capsid genes are. We could figure out the antigenic sites. They would be, that could be done.
But the problem is you have to grow them up in a permissive host. And that's the problem. It's not an E. coli fermenter. It's a mycobacterial fermenter.
Got it.
Right.
But you could identify, let's say, 10 antigenic sites on the capsid.
Yeah.
You could change each of so that it's no longer neutralized, and then keep those in store for the next round. So it's doable. So I would say it doesn't kill this approach, but we have ways that we can get around it, not just immunosuppressing, but, modifying the phages, which they can certainly do.
And
Yes.
it's also fair to say, isn't it, Vincent, that this is not a surprising result, but it's the
Not at all.
first time that somebody has really systematically analyzed it in a patient?
Yeah, I think that this, previously was done in either an immunosuppressed person or someone who, or they do topical treatments where it's not an
Right.
issue. But this is the first time, and it's not surprising, but it had to be done, right?
Yeah, and they got useful information and
Yeah, for
kind of
sure.
changed our focus.
Yep.
I was going to say it took 12 authors to do this work. It's relatively simple work. It doesn't take 12 authors to do this.
Oh,
This is,
let me talk about this interdisciplinary project, and then we'll come back to you. So I mentioned one of the three first authors is Dr. Rebecca Dietrich. She is a research associate in Pittsburgh in Graham Helpfell's group. She distinctly remembers sitting in seventh grade science class and deciding that she loves science. She doesn't remember what it was they were learning, but she just loved the challenge. So she went to Slippery Rock and majored in biology, minored in chemistry, and did some research in a botany lab. But then she took a class in microbiology and molecular biology with Steve Strain. And she also was doing some work in his lab, preparing media, et cetera. And he basically mentored her as she chose to apply to PhD programs and then went on to get a PhD at Duquesne University in Pittsburgh. So she also married during that period and later was fortunate to get an interview and land a postdoc with Graham Hatfull, one of the senior authors, studying these mycobacteriophage. And she, as I mentioned, was the first author on that Nature Medicine 2019 report, which was the first successful application of phage therapy in a patient. So since that paper was published, they have received from all over the world 200 different Mycobacterium abscessus clinical isolates. So they are building not only a huge catalog of phage from their C-phage project, but also these mycobacterium. So it was her job, along with Bailey Smith, a technician in the Hatfill lab, to screen the clinical isolate for this particular patient to identify in their large collection of phages which ones could wipe out the mycobacterium. And she remembers just how exciting their first results were when they saw that the sputum counts of the mycobacterium had been decreasing. But then, as we learned from Vincent, after a couple of months, the celebrations stopped. She got a sinking feeling in her stomach, realizing that the neutralizing antibodies were really a problem, and then collaborated with Krista Freeman, another postdoc in the lab, to really quantify the immune response and make clear what the blockade was. So she also has 2 children and says that while being a wife, a mother, and a scientist is difficult, her family is her priority. She loves the ability, though, to do science that has the ability or potential to impact people during really tough moments. And this is personal for her because when she was a first-year college student, her father was diagnosed with a cancer. They were able to put that in remission, but she has since lost her dad to that cancer. So it is a personal drive of her to try to do science that can help people and their families. So her colleagues on this paper were another doctor who's doing a postdoc with Graham Hatfull, and that's Krista Freeman. She got her bachelor's in physics with a minor in mathematics from Cleveland State University, and then got a master's in physics at Carnegie Mellon. And her PhD was also in physics. And Vincent, she was studying viral DNA packaging and ejection controlled by capsid stability. So a physics project.
Cool.
But her postdoc with Graham Hatfull, of course, is allowing her to now take this to a new level. And she says that with her background in physics and biophysics, she initially was studying polymers that could be used for drug delivery. So there's a little bit of overlap here. And then with her use of viruses during her graduate work, this postdoc with Graham Hatfull was a really great application of her skills. And she also said there was a silver lining to COVID because during the pandemic, she and her colleagues in the Hatfull Lab started exploring using phage-based vaccine development. So that's where she learned a number of the techniques, the ELISAs, the Western blotting, that came to use when others in the lab approached her about joining them on this particular patient who they wanted to apply phage therapy. So she also really admires Dr. Kira Cohen, the physician who cares for the patients that they have treated in these two papers, and also does remember when this project really got cooking. The experience basically took over her life. So, for example, she remembers just having such a sense of urgency to get the results. She couldn't pull away from herself away from the lab. And she remembers developing a blot one Saturday night at midnight to see what was happening because the sense of anticipation was so strong. She said it was fast-paced and thrilling. But her advice for other colleagues is never be afraid to learn something new. With enough determination, plus support and collaboration, you can make a meaningful contribution to any problem in science. Now, they also worked with Jan Nguyen from Johns Hopkins, and she got her start actually as a music performance major, with also some science on the side. And she really loved the collaborative nature and universal presence of both fields. So later she decided to focus more on science. She got a master's at Johns Hopkins and also did a postbac, was a fellow at the National Institutes of Health, and then joined Johns Hopkins as the first team member in this new research program focused on the non-tuberculosis mycobacterium. So it's her job now as her title is a research program manager. She worked her way up the ranks from a technician. She became a senior research program coordinator or research program manager. So she manages all their translational research program and oversees a team of colleagues, including she wants to give special credit to Ashley Budhartely Talbot and Dr. Lisa Ruppel, who Ashley, for example, has very careful hands, critical reasoning, meticulous microbiological technique, and she's the one that performed the quantitative mycobacteriology to get the phage efficiency numbers that went into this paper. And Ashley also manages all aspects of the wet lab and is a key part of the team. And then Dr. Lisa Ruppel is the team's research pharmacist. So you can see that with a patient's life at stake, they bring in specialists from many different disciplines to oversee this project and the progress of the patient. So she's the one that really admires the bravery of the phage patient and said it's just been a real privilege to work with them. And when Jan is not working, she loves spending time outdoors with her husband and their dog, and especially appreciates that her husband has supported her all through her academic and professional career, and he's been a real partner. She closes by saying that our generation of scientists are part of an era of novel therapeutics. We have the potential to overcome many challenges of the non-tuberculoid Mycobacterium tuberculosis. And she hopes our generation continues to pursue translational research through innovative, thoughtful, and collaborative approach.
Nice.
So those are just three of the people that contributed to this really important work, although sobering in some ways.
Yeah, wonderful. Thank you, Michele.
Yeah.
So for those of you who want to find out more about this phage program of Graham Hatfull, we interviewed him on TWiM number 87 some time ago, so check that out. All right, that'll do it for TWiM 247. You can find the show notes at microbe.tv/TWiM. Send your comments and questions to twim@microbe.tv. And if you like what we do, consider supporting us, microbe.tv/contribute. Michele Swanson's at the University of Michigan. Thanks, Michele.
It was a pleasure. Good to be with you all.
Elio Schachter's at Small Things Considered. Thank you, Elio.
Oh, wonderful. Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thanks, Michael.
Thanks, Vincent.
And I am Vincent Racaniello. You can find me at virology.com. TWiM.blog. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.